Hi, my name is Riley Kent, and I am working at the Institute for Systems Biology in Seattle, Washington for the summer. And what I have behind me and what we are focusing on for this project is related to the inflammasome and inflammatory disorders within the body. So specifically with the inflammasome, we are looking at the proteins called NLRP1 and NLRC4, and what we have noticed is sometimes these proteins will create clumps or clusters within the cell, and not much is known on why they do this process. So this project is going to be looking at why the cells might be doing this process and if there's any importance for the cell and for that immune response. When these proteins are overexpressed, they will induce inflammation and likely they will cause cell death. So this leads to a lot of different autoimmune disorders, cancers, and other diseases within the body. So we're focused on why these clusters happen and if there's any link to cancer, autoimmune disorders, metabolic disorders, anything of that sort. So in this video, I will show you how we actually get pictures like the ones behind me here, how we prepare those slides, how we culture cells for this project, and much, much more. So I'm excited to take you along and show you this process with me. So here is part of our culture room, and this is the station that I will be using today. What I am doing is I'm taking my cell lines that I have been looking at for this project, and I will be splitting them. So I will be taking the media inside of those tubes and replacing it with some new media and putting our cells back into those flasks. In a little bit I will show you what those cells actually look like. This process that I'm doing today is really important and it helps us maintain the cultures and keep them for long term. So what I went ahead and did now is I took our media and our cells out of these flasks and I put them into these tubes so we could centrifuge them. And if we take one of these tubes, you can actually see the collection of cells down at the bottom there. And those cells will be diluted from these tubes and put back into the flasks so we can store these long term. If we don't do that, they'll continue to grow and eat up all of our media. We don't want that to happen, so we need to take them out of our flasks and replace them with a diluted sample. Now, in the meantime, I'm going to take some of these cells and place them into our slides so that we can look at them under the confocal microscope. Now, the next part of this video will show you some of those cells under the confocal microscope, and you will get to see what our project is actually getting at. So now that I showed you the process of culturing our cells and maintaining those cell lines, I went ahead and prepared a slide for you to see in this video, and I have it already processing on the Now that I showed you the process of maintaining our cell culture lines, I went ahead and created a cell slide for you to see on this microscope, and I already have it running here. So this is our confocal microscope, and I will actually pick you up and show you what's going on. This microscope here is able to shine a few different lasers onto that cell or onto those groups of cells. And we can use this program on the computer to basically image different parts of that protein that are being expressed. So here we have 3 different parts of the protein being expressed, and this corner is overlaying that data. So you can see in some of these pieces here, those dots are those clusters that we are concerned about for this project.